Hello, everyone, and welcome to today's keynote presentation, Multi-Organ Human on a Chip Systems for Preclinical Efficacy and Toxicity Evaluations. This webinar is part of the Cell Biology virtual event, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them in to the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of the screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab link located at the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. James Hickman, co-founder and chief scientist of Hesperos, founder director of NanoScience Technology Center, professor of Nanoscience Technology, Chemistry, and the School of Medicine, and the Electrical Engineering at the University of Central Florida. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Dr. Hickman, welcome. You may now begin your presentation, sir.
Well, thank you much for the invite, and thank you much for the kind introduction. And let me see how to get this set up here. All right. So, can you see the presentation?
Yes.
Wonderful. All right. So, just give you my conflict of interest. I am also co-founder with Michael Schuler of Hesperos. We were founded in 2015. We've licensed a number of patents from Cornell and UCF. We both won the Lush Prize for alternatives to animal testing. The company has used a lot of SBR grants to develop the technology. We're a contract research organization in a 14,000 square foot facility. We graduated out of the UCF incubator about 3 years ago, about 40 people. And I'll talk to this about this in the during the presentation, but we've now shown that we can take a microphysiological system and for efficacy, combine it with phase 1 safety data for another indication. And we've gotten the allowance into a phase 2 clinical trial for a repurposed drug, which is a huge milestone for these types of technology. So I've been working in this area for about 25, 30 years, which just means I'm old. But it also, we started this idea of clinically relevant functional readouts a while ago. And what does that mean? It means, well, you walk into a doctor's office, he doesn't immediately stick a needle in your arm to be able to pull out fluids, look at biomarkers, right? He look, how are you walking? How are you talking? Are you making sense? He listens to your heart. He checks your reflexes. These are all clinically relevant functional readouts because he can get information by doing all those very simple tests, non-invasive tests. We can do the same thing by monitoring muscle contraction, electrical activity from neurons and cardiac cells, be able to look at neuromuscular junction formation, which is a motor neuron innervating a muscle. Also look in terms of your barrier integrity, in terms of your GI tract, in terms of your blood-brain barrier, in terms of your kidney function. These are all what we call clinically relevant functional readouts. So it allows us to do about 95% of your literature is looking at acute as dosed. And what we try to do is to look at this, use these non-invasive techniques to be able to look at chronic monitoring of these systems. And it can also allow for mechanistic determination. I'll talk about that later on. And really what this idea is, we can take and put neurons over microelectrodes to be able to get electrical signals. We can pattern cardiomyocytes over microelectrodes to be able to look at conduction velocity, QT interval, beat frequency. Again, we can look at the cantilevers. We can take skeletal, take cardiac myocytes, put them on top of these thin pieces of silicon. So when they contract, it bends. We can bounce a laser off the bottom, see the deflection, and be able to translate it into force. Do the same thing with skeletal muscle. We can still look at biomarkers. We can monitor liver function on a daily basis, and we can also use TEER to monitor kidney and other barrier function. So I was the first to create a totally defined cell culture system back in 1995. For what we use self-assembled monolayers. We could then put proteins and peptides on top of them. We can make geometric patterns. I banned the use of serum in my lab about 25 years ago. So everything I'm showing you is all serum-free media, even in the multi-organ systems. We've done adult, embryonic, rat, mouse, and human. I'm going to focus on human today. Cell preparation is very important, and it really hasn't been a cell type we haven't been able to culture with these systems. I'm going to start off with the neuronal-based models. I drew this cartoon about 15, 20 years ago to be able to start trying to understand learning and memory. Then the fundamental unit of learning memory is something called long-term potentiation. And my idea is to look how hippocampal cells communicate with cortical neurons and how they store information using a combination of microelectrodes and neuron connections. And so the first thing you have to do is be able to get functionally mature neurons. So we've differentiated in our lab mature cortical neurons from induced pluripotent stem cells. You can see the correct markers for this. And what you can see is that early on, you get a mixed population that goes into a mostly cortical population after about 28 days. At that time, we basically follow it in terms of electrical maturity as well, looking at spontaneous activity, basically looking at current clamp, electrical activity as well. And you'll be able to see nice sodium potassium currents and be able to show that the activity goes up with age, and also that the resting membrane potential, percentage of firing cells also goes up as well. Now, what we can do is we want to try and create patterns of these systems so we can isolate which neurons are talking to each other. So we use this self-assembled monolayer chemistry to modify the surface. It's based about a nanometer or so thick of a monolayer. Where we could use a trimethoxy, monomethoxy, trichloro, monochlorosilane, where this R group can be anything that we can synthesize and attach to a silicon group. And then we can put a mask over it, shine light on it, and then come back with a second molecule to make a pattern. This is what one of the patterns looks like, where we're trying to put a somal adhesion site here, and then dendrites here and axons here. These are two neuronal cell bodies here. They've just been plated onto the surface, not placed in any way. They put out a process, a pathfinding process, that finds the somal adhesion site. And then there's communication back and forth between the process and the cell body. It says, okay, it's okay to move now. It's a good site to move to. And then it puts out the neurites to become the axons and the dendrites. And we can control that all with surface chemistry. You can see here the axon going out and the axon going out on this side to be able to form the two-cell circuit. Now, we can also do that with populations of neurons. This is a population of cortical cells and another population of cortical cells, which are attached through the axonal tracts you can see here. And because these are isolated from each other, these are all 5 separate experiments, assuming that they are connecting properly. Again, you can see the phase images here. Again, you see the correct markers for the system. Now, very, very important. It's real easy to mistake noise from a microelectrode system with electrical neuronal activity. So what we do is we can actually take the signals that we collect, hit it with lidocaine, and we basically know that we abolish those signals. It's not noise, it's actually electrical signals, and can confirm that. Again, I mentioned the maturation is very important. We can use a measure of long-term potentiation to show that the Immature, we cannot induce long-term potentiation, so these are not functionally mature neurons at 21 days. However, we do know that it's a mixed population of inhibitory excitatory neurons, and if we block the inhibitory neurons using picrotoxin, we can induce LTP. And we can also use this as an epilepsy model. But we actually show after about 35 days in culture, we don't need the picrotoxin. Most of the neurons convert to excitatory, and we can now do long-term potentiation in these systems. And just to show that it really is LTP going through synapses, we can stain for synaptophysin and glutamic 1 receptor and show that they're both present. We can also dose it with the AMPA receptor inhibition and show we can abolish the signals. Same thing with an NMDA receptor. Again, you cannot do long-term potentiation without an NMDA receptor function. Again, as we then treat this with increasing, basically treat this with system, you see that the you can then dose it and then see that the electrode activity goes down. And again, showing that we have lost the activity when we block the NMDA receptor. So we really are doing long-term potentiation. We can then use that model to be able to look at the two main factors in Alzheimer's disease, A-beta oligomers. In this case, we're looking at A-beta scrambled, which is the A-beta peptide, but we scrambled the amino acids in it. And basically, so we can maintain LTP. We can see actually we can induce LTP, then treat it with the oligomers, and we abolish that completely. We can do the same thing with tau oligomers. So basically, we can maintain LTP, and it is abolished. This was published in Alzheimer's and dementia. Okay, and again, just showing again to show we get really good equivalence between the patch clamp electrophysiology with these systems. This is patch clamp for the A-beta scrambled. You know, basically seeing induced activity and spontaneous activity. When we treat it with the oligomers, this is mostly goes away. But we then treat with A-beta with a drug, okay, we can actually bring back most of that activity. And again, this mirrors the MEA electrical activity, mirrors the patch clamp. So we know we have a really good, valid model for LTP. And again, really important, we're doing this all without cell death, okay? Very, very important. If you're trying to create a clinically relevant functional readout, okay, if you've got cells dying, you're in trouble if you're in a clinical trial. And so in this case, we're doing this without cell death. We're just looking at functional changes. In the case, again, we can look at the A-beta scrambled, A-beta 42, and then treat it with donepezil, which is an approved AD drug, to basically show that we maintain the long-term potentiation. We've now looked at the not only the two approved drugs, but two off-target drugs being used for AD, and showing that all of them we can actually treat with A-beta scrambled, A-beta oligomers, and then be able to show we can retain activity for donepezil, scaratinib, memantine, and rolopram. Basically showing that this is a really good model for being able to screen for A-beta Alzheimer's therapeutics. But again, because we don't have cell death, we can actually mimic the mild cognitive impairment or before that aspect of Alzheimer's, because after you get full-blown Alzheimer's, you get significant amounts of cell death. We're doing this with low enough concentrations, seeing an attenuation of function without cell death. And again, just one other test that we wanted to do. We wanted to show that we could stimulate on one set of electrodes and then record on the other, and then be able to show that we can have then the non-stimulated control. We don't see any LTP, but then when we stimulate on one electrode and look at the other electrode, we see that we can induce LTP. So we know we can actually are doing this through synaptic activity. And again, we can compare the two panels of interest. Why that is important is because one of the things I did do earlier is Tom Bowen and I invented bioprinting when we were both at Clemson in the early 2000s. And so if you combine bioprinting with our surface patterning, we can actually put down the same population of neurons, but two different populations of neurons. So if I want to look at, say, Parkinson's, I could put a dopaminergic neuron here and a cortical neuron or another neuron here, and to be able to then look at the the circuits of a mixed neuronal population, which opens up a huge number of neuropsychiatric diseases, conditions that we could potentially look at with these models. All right, so I'm going to move on from the CNS model to look at a PNS model we've developed. You can see here, we can actually make a chamber where we can take motor neurons, okay, and put them in one side of the chamber. You see these small little tunnels here. We can then grow axons through those tunnels to innervate muscle on the other side to be able to look at neuromuscular junction. Again, one of those tests that a doctor does in the office when he whacks you on your knee, he's looking to see whether or not, first, whether the sensory portion is working by stretching the tendon, patellar tendon, but then that informs the motor neuron to then kick, okay? And that's the second half of the reflex function that he's looking for. So we can look at that. Again, we can stimulate using electrodes on one side. We can grow the myotubes on the cantilevers, or we can look at motion capture here. Again, looking at axons going through the tunnels, making neuromuscular junctions on the other side. One of the first things we did was do a dose response curve for Botox, where it was very, very similar to the IC50 that had been determined by the company for this allergen, and which was done with an animal. So we have very good compliance in terms of the human model for this. And this was published in Biomaterials in 2018. However, really looking for this in terms of disease modeling, ALS, myotrophic lateral sclerosis, or Lou Gehrig's disease. We've been able to take patient samples, induce pluripotent stem cells for a number of SAD1 mutants, FUS, TDP43, and C9. We can then differentiate out those mutants, put them in the motor neuron chamber, grow axons through to then innervate the muscle. And again, this is an example of motion capture, where again, these are the stimulation pulses, and these are contractions. We can marry them up and see for wild type, we get good fidelity. For the ALS mutation, we don't, but then we can then treat it with a therapeutic and be able to restore some of that function. And again, getting to this whole idea of a clinically relevant functional readout. Before we showed long-term potentiation, in which has a correlate for learning and memory. In this particular case, we asked somebody who was doing an ALS clinical trial, well, what are some of the measures you have them do? I said, well, one of them is they have somebody do a task with their hands faster and faster. And what they have to find out is that as they do it faster and faster, they start getting jittery and getting spastic, and they lose grip strength. Well, so we can reproduce that. We can take and stimulate the motor neurons faster and faster, and eventually drive it into tetanus. And if there's any loss of contractions here, that would be equivalent to the spasticity or the jittering. And if we see a loss of tetanus, that would be equivalent to the loss of grip strength. You can see here, this is a very busy slide, but you can see here for the wild type, we get a nice fidelity of the system. For one SOD1 mutation, you get some skipping, partial tetanus. For another ALS mutation for SOD1, you see some skipping and a complete loss of tetanus. And for the FUS mutation, again, you see some fidelity, but you see that lost period of time, and you can see no tetanus in the system. We have a control in the system, in that we can look at the muscle directly by stimulating the muscle. You can see here, and we see very good fidelity, which means that the deficit is at the neuromuscular junction. We can then monitor this for all the different mutations. You see the deficits as we increase higher and higher. We can also look at the fatigue index. Again, this is another clinically relevant functional readout that they do. And see the fatigue index goes up for the mutations. And also you can see that we then treat it with what's called the Deanna protocol, which is a holistic treatment created for his daughter by a physician. Her name is Deanna, which anecdotally has shown over about 1,000 people using this on his website, showing some benefits of delaying loss of ability. We can see we can reverse the fidelity with that treatment. And also, not so much at day 14, but after a longer treatment period, we get to see a big change in terms of the fatigue index, which is, pretty much bring it back to baseline, okay, with treatment. And this was published in Advanced Therapeutics in 2020. We can also use the same system to look at myasthenia gravis. Myasthenia gravis is a disease where your body creates antibodies to the acetylcholine receptors on your muscle. So when the nerve terminal releases acetylcholine, it can't bind to the receptors on the muscle, so the muscle will not contract. So you can actually have a blocking effect. You can have it where it causes an internalization, or you can have complement deposition. Be able to do, again, the first dose response curve for a biologic in this system. And again, we show this for the acetylcholine antibody. And again, another antibody didn't have any effect. And basically, you see for the muscle itself, we have a nice area of here where we don't see much effect, but even in affect the muscle by itself at really high concentrations. So we can use that idea and then basically look at the acetylcholine receptor for the untreated. And so we have about the same amount on both externally and internally. We treat it with 2 micromolar of the antibody. We show almost a complete abolishment of the acetylcholine receptor on the outside. It's still present on the inside. We go to a higher concentration, we can see a down regulation of that receptor. And we can also show that there's complement deposition and we can actually have seen all 3 mechanisms in myasthenia gravis, and that was published last year, I believe. We've also been able to show that for myelination, we can take and myelinate the motor neurons. This was the first human Schwann cell and human motor neuron publication showing pretty good efficiency in the model. And so somebody had seen that, a company called True North, which was acquired by Biography, which was acquired by Sanofi. And they were interested in looking at conduction velocity loss for 2 rare diseases, multifocal motor neuropathy and complement-induced demyelinating polyneuropathy. And because what happens here is the antibodies are raised and start attacking the nosoronviae, okay, because it activates the complement cascade. You can see here in vivo aspects where you're getting, just for a ENG measure. I've had this done before. It's not a ton of fun. And basically, what you stimulate the nerve, and you look and see the conduction velocity. You can see that this is lost with time. And they said, well, okay, can you create a motor neuron Schwann cell system to be able to look at complement deposition? So we took patient sera samples and we got a large amount of complement deposition and showed that their drug abolished it. But again, that's a nice correlation, but it's not getting to this clinically relevant aspect to it. So we took our NMJ system, turned it 90 degrees, and now put the electrodes in the tunnels and grew axons over those tunnels. We can then follow the conduction velocity. And since this is a measure that people use clinically for these diseases, we then were able to take in the untreated system, show that we have a range of frequencies, and then take the serum control for the patient and the isotype control. And again, before we treated it, we see a nice range of conduction velocities. And then the before with the serum with their molecule, and show the control, we were fine. But with the patient sera and the isotype control, conduction velocity was abolished. Whereas with their molecule, it was retained. Now, this data, okay, was the only efficacy data used, as I indicated before, okay, for an IND that was filed by Sanofi using safety data for another indication, okay, which was allowed to proceed into a phase 2 clinical trial for repurposing the drug for CIDP. Okay, and this is the clinical trial number. It was started recruiting patients in April of 2021. And again, this was published in Advanced Therapeutics, all the data we used for that, which is a huge deal because now one of the aspects of this, which is the regulatory acceptance, has been shown, at least in a limited sense, for a rare disease. So now I'm going to move on from the neuronal systems to be able to show you that we can take these models and combine them with other organ systems, okay, besides just muscle. And we do that based upon work that my collaborator, Michael Schuler, did, where he took the physiologically relevant pharmacokinetic models, okay, that were used to determine phase 1 dosing in a clinical trial, modify those, and use it to design the very first multi-organ chip back in the 90s. We've now taken this to the next level, and we've been able to look at the for opioid overdose. We were able to differentiate out pre-BOTC neurons. Pre-BOTC neurons are those that control the rhythmic breathing for their lungs, and that is actually the main target for when you overdose, is those neurons shut down. So we're able to take and shut those down with the opioids, but then recover with naloxone in this multi-organ system. But at the same time, we're monitoring the off-target toxicity, looking at liver, cardiac, kidney systems, okay? Because what they're finding is for addicts who have been repeat dosed with naloxone, they start seeing some deficits in function for cardiac and kidney. Now, what makes a lot of this possible is the idea of a pumpless platform. Mike invented this back in 2010. This is the cover of Lab on a Chip. And what we basically can do is we use gravity to pump it. Instead of using peristaltic pumps or something else like that, it gets rid of the tubing, the pumps, and it also gets rid of the bubbles, which is if you use these systems, it's an extremely difficult problem. But also it makes it a low volume system, so I can see the effect of not only parent but metabolite in the same system. So just be able to show that we've been able to differentiate the pre-BOTC neurons. These are all the appropriate markers for it. Basically showing the main one is the NK1 receptor and the mu opioid receptor on these neurons. We then showed them functionally. We could then treat them with ATP and stimulate them, substance P, stimulate them, and looking at somatostatin and showed that it actually turned it off. And these are the main functional aspects for pre-BOTC neurons. So we actually have the functional aspects for looking at overdose, again, without cell death in these systems. We're able to use those models to be able to get the IC50s for Damgo, which is a synthetic opioid, methadone, fentanyl, and codeine. And these agreed fairly well with the literature values that you find. We were then be able to take those systems and look at monitor the electrical activity, shut it down with the opioids, and then get recovery with naloxone in these systems. We've also been able to differentiate nociceptors to be able to look at pain. Nociceptors are some of the neurons where you have the axons located close to your skin, which are pain is one of the things that they sense. Again, be able to show that we can differentiate out the nociceptors, that we can then have the correct markers for that. This was recently accepted for publication. We can then take those nociceptors and then be able to treat them with ATP to turn them on, and then treat it with lidocaine to shut them off, and be able to do this in a dose response manner on the microelectrode arrays as well. So we've kind of showed you a wide variety of different neuronal systems we can use in these human on a chip systems, both single organ, but also multi-organ. Now I'm going to then get more into the multi-organ aspects of it. And I've mentioned the cardiac function a number of times. Just going to show you this is how we make them. We can actually take a laser and shine it on the pattern and be able to transfer the pattern and line it up with these microelectrodes. So you can see the cardiac cells over top of microelectrodes and get rhythm generation, conduction velocity, AP length. But on a separate chip, we can actually then, again, grow the cardiac cells on these cantilevers. So when they contract, it bends, it deflects the laser. We can then get force out of this. And this combination was published in Biomaterials back in 2015. So what we can then do, we can then monitor the conduction velocity both spontaneously, but more importantly, we can stimulate it. And we can follow it over stimulate at this electrode, follow it over these electrodes. We can have a gap and pick it back up again. And we can also measure QT interval. QT interval is very important because that is one of the things that is prevents drugs from going into clinical trials, but it's also the main reason for aftermarket removal of drugs that had been previously approved. We've done well over 50 drugs with this system. I'm just going to highlight 3. You can take norepinephrine, speed up the heart. You can do it in the dose response manner. We can take verapamil and shut down attenuate the force. We can take sotalol, and so it has no effect on force, but it causes a long elongation of the QT interval. Okay, and what happens with that is, when you're firing action potentials for the cardiac cells to drive it, if this gets too long, it starts interfering with the next action potential, which causes fibrillation, which is why the QT interval is so important and so deadly. And again, this is all done without killing the cells, because again, when your heart goes in fibrillation, it stops pumping. You don't necessarily could kill any of the cells in the heart, but if it stops functioning electrically, then you can shut down its function, and then you'll kill the organism. And also, we can then say if we're looking at this as a phenotypic model, which is we're just trying to reproduce a deficit without really knowing what the target is, but you want to then know what the target is, we can say, well, okay, all the pathways involved in force, okay, not affected, and we can then focus on the electrical pathways, and then start doing selective blocking, like I showed earlier, with the AMPA receptors and NMDA receptors, and be able to start then isolate what pathways might be involved for a particular drug. We can then take that heart system and then combine it with a liver module we've developed and create a heart-liver multi-organ system. And we did some work with AstraZeneca, where they had a molecule they couldn't figure out why they were getting deficits for it in the animal model when it was clean in the in vitro assays. So we were able to solve that problem for them. But at first they wanted us to validate the model with a terifenadine-fexofenadine couple. Terifenadine is an antihistamine that was pulled off the market because it caused QT elongation that was fatal for some patients. However, its metabolite, fexofenadine, which is now Allegra, has no indication. So we said in a cardiac-only system, okay, we're able to see the QT interval was shifted far to the left, but when we added the liver, it was shifted far to the right within the safe zone. Conduction velocity was not affected with the liver present, but with only cardiac, we had an IC50 of 33 micromolar, okay, for the terifenadine. And again, cardiac viability was not affected with these systems. Now, this becomes very important. Say if you're a medicinal chemist, you're trying to work with 2 or 3 different combinations of your lead or derivatives of your lead compound, and you were worried about metabolites, you could just take our system and put your molecule in it, and we'd be able to tell you whether or not you had a metabolism problem or not in terms of toxicity, in terms of cardiac, by just simply doing it with and without the liver present. Okay, and you wouldn't have to identify the metabolites up front. Okay, you would say, okay, this is a fairly clean drug before you go into an animal model, and you wouldn't waste a whole lot of money on animal models and also scaling up, which is one of the key drivers for cost for therapeutics, which is that scale up to going from milligrams to kilograms of compound to go into an animal model. Now, we can also create models of these systems in silico. We grid the system up. We can actually look at the flow rates in the system. We can tune the shear stress. We can also look at the mixing. We can also then account for the loss in terms of metabolism, but also any absorption into the housing materials. And again, some materials absorb a lot, like terifenadine, some not very much, like fexofenadine. But what we really want to try to do is calculate the area under the curve, okay, because that's very important, because 95% of the literature is as dosed and don't account for any losses that they get with the molecule. And if you're going to try to be able to, predict something in vivo, you would going to need the area under the curve as opposed to the as dose concentration. And we can then take this in silico models we create, show for repeat dosing. We get a very nice curve for the metabolite and for the parent compound. We can measure this in the system using HPLC-MS and show it's a good correlation between the in silico predictions and the actual measurements. We use that to be able to create a pharmacokinetic pharmacodynamic model where we can account for absorption, be able to do the pharmacokinetics for that, account for metabolism, create the pharmacokinetics for that, and then be able to take those pharmacokinetics and then our exposure curve to be able to determine the pharmacodynamics both for the treatment on the outside, but also then account for transport inside the cell. And again, for the hERG channel, which is one of the main reasons for QT elongation, you want to be able to get the PD for that because that's internally accessed, and you get the PD for that. So you can account for that transport across it. And this is published in Nature Scientific Reports in collaboration with AstraZeneca. And again, what we're able to then do is take that in vitro PK/PD model, be able to create an in vivo PK/PD model to use that data to predict guinea pig, dog, and non-human primate data as well. We also collaborated with Roche with the systems. And he said, well, that's a really nice model of toxicity. Can you do efficacy and tox at the same time? And so they gave us a fairly complex problem, which is to look at a multi-drug resistant cancer and a non-multi-drug resistant cancer to be able to look at efficacy and off-target tox in the system. And they chose tamoxifen. Tamoxifen by itself is not a very good chemotherapeutic. However, metabolite is. So tamoxifen by itself really didn't decrease the cancer proliferation, but with liver present, it did. Its metabolite by itself also decreased proliferation. However, in the multi-drug resistant cancer, you can see with no liver, no effect, with liver, no effect, with the metabolite, no effect. But we added a second drug to block the PGP channel. So the PGP channel in cancer cells basically pumps out the chemotherapeutic as fast as it goes in. So by blocking that channel, we are now able to see a decrease in proliferation. And we didn't see any increased decrease in proliferation, showing it was blocking the channel. It wasn't acting as a chemotherapeutic by itself. But we were able to then monitor the off-target tox at the same time of the cardiac cells in terms of conduction velocity, which went down with tamoxifen but recovered. Force went down but recovered. Beat frequency went down a little bit and recovered. But for the drug-drug combination, it went down but did not recover. Force went down and kind of did recover a little bit. Beat frequency came down. But again, we haven't killed any of the cells here, but you would have basically killed the patient in this drug-drug combination. So what that data would tell you, you had to wait longer before you would have the second dose, or you had to replace this combination, maybe replace verapamil with something else. And again, showing that liver function in terms of albumin production, urea production was maintained, and obviously the metabolic function was maintained because we saw the aspect of the therapeutics. And this was published in Science Translational Medicine and actually featured in Francis Collins's blog, saying that this is a very important advance for the field. So again, we can take those neuronal systems, muscle systems, cardiac system, liver, put them together in a multi-organ system. We published the first multi-organ recirculating system with AstraZeneca, with L'Oreal, back in 2016. We're able to look at 5 different drugs, and we're able to reproduce the toxicity expected for certain organs, but also didn't show any tox for other organs that we didn't expect it for it. We also, in 2019, published L'Oreal taking this multi-organ system out for 28 days, be able to monitor it functionally. That's a very important time frame because most basic toxicity experiments with animals go for 28 days. And it's just showing you that the morphology of the liver is maintained for 4 weeks. Albumin production in the multi-organ system is very stable. By itself, it's not. And again, in static, you don't see it. You see the loss of activity quite quickly. And these are just 3 literature values that we compared it to. Urea production, and then CYP activity is maintained. The cardiac cells are maintained on the cantilevers. again for 28 days. Very good maintenance of function. You see basically the electrical activity, again, beat frequency, QT interval, conduction velocity is maintained. You see the patterns are maintained for 28 days. You can see the skeletal muscle. You see the myotubes still in the cantilevers. And again, must stable function for 28 days. Then we can then pattern motor neurons over the microelectrode as well and show stable function for 28 days. We are also working with NIEHS. They have rat systems where we can create a bridge between in vivo rat data and be able to rat on the chip, and then also then be able to compare that to the human on the chip data. We've also been able to create an innate immune system on the chip. This was done again in collaboration with Roche. This is published in Advanced Science, where we're able to take monocytes, and you can see them flowing in the system. And what we were able to show for 7 days, we could flow the monocytes without activating them. And again, you can see that the markers for activation do not come up. However, we can treat them with IL-4, okay, and you see that these floating monocytes then turn into macrophages, adherent macrophages on the surface. But we really wanted to get to the granular with the system. Can we actually reproduce the restorative or the M2 phenotype reaction as well as the inflammatory or M1 phenotype? So we then selectively damage the cardiac cells with amiodarone and be able to show that after a period of time, you get progressive infiltration after treating it with amiodarone of the monocytes. You see them basically attaching it, infiltrating into the system. And you can see here that the amiodarone does cause a loss of function, but this was exacerbated by the presence of the THP cells, and you did not get recovery. For the force, conduction velocity went down, but did recover. You see the beat frequency went down and recovered to some degree. So it's really going after the electrical activity and not as much the force, but you see that the muscle force was not affected and the liver function was not affected. Now, if you then look at the cytokine profiles, you see IL-6 goes up and is maintained up, which is a signature for the M2 phenotype. And we see all the cytokines for the inflammatory phenotype are not upregulated. But again, we can actually then cause the inflammatory phenotype by treating it with interferon gamma and LPS bacterial protein, and showing that the IL-8 production goes up. And again, we can see that if we just have the monocytes and then treating it with the system first with the LPS and the interferon gamma, there's no effect. The presence of the monocytes, you see the cardiac force go down. Conduction velocity really isn't that much changed in the system. Again, electrical activity, but you see that the force of the skeletal muscle is also compromised, and the activity of the liver was also compromised, showing that what we're getting is all the organs are being attacked at the same time. And you see IL-6 goes up, but then comes down. We see IL-8, IL-10, RANTES, MIP-1, TNF-alpha, NCP-1 are all upregulated and maintained in the system. We can also integrate barrier tissues into the models. Obviously, working with L'Oreal, we were able to create a 4-organ plus skin to create a 5-organ system. We can, again, as I showed you, add kidney, but not for elimination, but this is for just as a tox target. Mike Schuler, my partner, had published a single-organ blood-brain barrier system, where again maintaining TIR for a long period of time, and also showing that transport was similar for on chip to in vivo. You know, this would, if this is a 45-degree line, this would be a perfect correlation. But you see, it's actually pretty good for caffeine, cimetidine, doxorubicin, and then increasing molecular weight dextrans. Also, he published for GI tract in terms of now the TIR value was lower for BBB, but you get very good barrier formation in the system. Now we're working right now to create a 3-organ GI tract BBB CNS model where we can actually treat with oral, IV, or intrathecal, which is directly into the CNS module. basically just showing that the nice tight junction information for the GI tract and with the proper markers for that, and also for the BBB. Tight junctions also maintained for that as well. And to show that the BBB transporters, GLUT1 and PGP, are present. And it's showing the electrical activity for the CNS patterns are also maintained. And then we're getting nice, good tier values for the GI tract and for the BBB in these modules. And again, we can actually look at the oral dose versus IV dose of caffeine. Caffeine, obviously, within a couple minutes of drinking your cup of coffee, you can feel it. Bicuculin also goes across both GI tract and IV very quickly. However, kyanic acid does not go across the CNS or the GI tract very well, but it does go across the BBB very well. We're able to reproduce those effects. And again, just reproducing the CNS LTP in the system. And lastly, you're saying, well, okay, you did get some correlations in terms of the regulatory acceptance. We had anticipated that early on. We started doing workshops on this idea of validation and qualification back in the 2012. We ran 6 of them. The 6th workshop was held a couple years ago on rare diseases. And pretty much what came out of there, which is, that one is that efficacy and safety are very, very different, okay? And so efficacy is for rare diseases, especially a good target for these systems. But what else could you use efficacy for, even if it wasn't a rare disease? You think about preclinical results to steer or limit clinical trial construction. Basically, come up with your companion diagnostics at the pre-animal stage, reduce eventually the number of people necessary for phase 3 clinical trials by creating human variants to create uncommon genome profiles. Because remember, a drug Vioxx passed the phase 3 clinical trial with 30,000 people, and still there was certain variants that came up that caused death due to QT prolongation in these systems. Again, in vitro clinical trials for rare diseases, we've already shown we can get a repurpose a drug for rare disease and get into phase 2 clinical trials with just efficacy one of these systems. Better evaluations for children, aged individuals, and again, for the cancer example, it was also for personalized medicine. So this is a combination of my group in front of Hesperos from the group at UCF and also the group at Hesperos. I'd like to thank them for all their contributions. I'd like to thank funding from NIH and other commercial aspects. And thank you for the invitation and for the audience for listening, and happy to take questions. And I'm going to stop sharing now.
And thank you, Dr. Hickman, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We already have some great questions coming in. Our first question, Dr. Hickman, can you talk a little bit about regulatory acceptance?
So I already mentioned for efficacy that we've already gotten one drug repurposed in the system. And actually, during one of these workshops, Jonathan Goldschmidt, who was head of rare diseases at the FDA at the time, basically told the field, he said, safety is going to be much harder because the problem with safety is people die, right? You get that wrong, people die. Whereas for efficacy, you just disappoint people. But really, nobody's going to die if your efficacy is not proven to be correct. So efficacy is, really the low-hanging fruit for these systems. But again, safety, I believe, is something that we can do eventually. We're already using it to, again, try to de-risk compounds before they ever go into animal models for safety. We're working with somebody to try and use these systems to replace one of the large animal models for safety. But the main thing to remember is that safety and efficacy are very, very different.
Thank you so much. And our next audience member wants to know, What are the benefits on multi-organ?
Well, there's lots of companies out there for looking at single organ systems, which have their benefit. But there's very few companies who are looking at multi-organ systems because they're harder, much harder. But if you can do it and get it recirculating, you can actually see for human-based systems how the different organs are interacting with each other. So when your liver is excreting something, you can see in real time what those effects are and the function of the other organs. Say if you have a pancreas on the chip, you can see, when you treat it with glucose, is insulin is being produced, and how's that affecting the other organs in the system. You can also, people try to understand what exosomes are doing for signaling. You can actually look at that without having to do a lot of detailed molecular biology. You can create a phenotypic model for these systems. And again, looking at going to that idea of phenotypic, somebody comes in with a rare disease, without having a target, you can take induced pluripotent stem cells, create the organs that are in deficit, and then the FDA will accept that, if you actually have organs that are interacting with each other in terms of a functional readout, which is why multi-organ systems are extremely important for the prediction of in vivo outcomes.
Thank you so much. And we have time for a couple of more questions. This next question is a two-part question. The audience member says, I assume this system cannot be used in a high throughput manner. So would it be used with lead compounds only, which is at the later stage in the drug discovery pipeline? And how difficult is it to work with this technology?
It is difficult to work with it, especially in the multi-organ systems, which is why when Mike and I created Hesperos, it's only as a CRO. Now, there are some systems, tissues, cells, a nice rudimentary two-organ system that is user-friendly, and people do use that quite a bit. Yes, and it generally is a high-content system. So normally how we think about this is you take your high-throughput, low information content screens, where you screen your 100,000 compound library, and then you create your 3 or 4 compounds that are most promising, and then you start working with us in terms of your high content aspects of it. And then, especially when you get to your lead compound, then you want to be able to try to look different variations of it. We can work with you to try and de-risk which one is the best candidate to scale up to go into your animal models for your safety initiations. Now, some aspects of the technology we are making high throughput. Some of the CNS models, we are increasing the throughput to be able to do that fairly quickly, but that's in the very early stages, whereas the high content screening is robust and is actually being used by many different groups at this point.
Thank you so much. And Dr. Hickman, we have time for one more question. Currently, is this technology used more for efficacy or toxicity testing? And what future applications do you envision?
Well, it's being used both for efficacy and safety because most companies and groups that came up were thinking about safety initially because that's where the majority of animal use is. So lots of groups are still working on this for safety. Many large pharmaceutical companies have set up these kinds of microphysiological systems in their safety organizations. Um, Efficacy is starting to come around because, there's 7,000 rare diseases. There's only 400 active research programs because there are no animal models for them. So this actually comes up as an opportunity, as we've shown already with demyelinating polyneuropathies. Greg Trusky's lab down at Duke is looking at Fabry's disease, and other models that are out there looking at rare diseases. So I think there's an equal weight, okay, for looking at efficacy and safety in these models.
Dr. Hickman, thank you so much for your presentation today. Would you like to provide our audience member with any closing remarks?
If you have been hesitant about using this technology, about considering something besides your animal models, I hope this presentation has convinced you. You should at least start exploring them. Not necessarily to totally replace your animal models, to make them at least more efficient. Okay. And also, again, for efficacy, if you've got an untractable efficacy problem, these models can be used. Again, one of the things we try to do is custom build models. Okay. And it's not just us, there's other companies out there. So, at the very least, hopefully, I've gotten some folks out there who are been working with that only animal models to at least consider the fact that this technology is getting mature enough to be a good alternative potentially, or actually a very nice complementary technique to work with animal models in the field of drug discovery and toxicity.
Thank you so much. And I want to thank you again for your time and for your important research, Dr. Hickman. And before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today. And those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 21st, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care and bye-bye.